Good morning and good afternoon, everyone. My name is Susan Mayo. I am a co-host uh, from my position at FDA. I'm one co-host of this ongoing webinar series for ASA Biopharm um, Safety Working Group, along with my colleague Michelle Zhang, who is co-hosting from the industry side of things. And we welcome you today to our next topic from our safety working group. Next slide, please. This safety working group has 3 work streams, and today the webinar that will be given with talks from Dr. Loke and Dr. Hendrickson, with a panel discussion with Dr. Lang, will be a combination of information from workstream 1, the interdisciplinary safety evaluation, specifically the aggregated safety assessment planning team that Barbara Hendrickson is one of the co-leads of, and workstream 3. I believe this is the design and analysis. Leanne, you can speak more to that, and perhaps Barbara will as well. This is being brought to you by the Communications and Outreach segment, which you see to the right of this slide. And so that's it for the, brought to you by type information. And we can go now to the next slide. Which shows here the agenda for today. I will now turn it over to Ranjita Sinval from AbbVie, who will be our kind of like guest host for our webinar today. Ranjita?
Thank you. And welcome, everybody. Good morning, good afternoon. I see lots of attendees. I'm expecting this to be a global attendee list, including our presenter, Dr. Loke, who's dialing in from UK. So welcome to today's session. We are diving into a pretty important topic, and it's regarding the transparency and completeness of safety data available in the public domain. Accurate safety data is the lifeline for everyone involved in healthcare, from agencies, providers, patients who need this information in making important treatment decisions, to pharmaceutical companies who are looking to develop the next generation of therapies. Benefit risk is an important paradigm with any new drug approval. And yet for decades, researchers have struggled with the inconsistent and incomplete reporting of safety data that obscures the true safety profile of the new medications. So today's session is going to be kicked off by Dr. Yoon Loke, who is faculty at the University of Anglia, Norwich, UK. He's trained in clinical pharmacology and internal medicine at the University of Oxford. Dr. Loke's research interests in drug safety have been substantially involved in conducting systematic reviews of adverse events, and he's contributed to the CONSORT as well as the PRISMA guidelines on reporting harms in a variety of publications. So Dr. Loke will be tracing the evolution of the safety reporting from the early challenges identified in the 1990s to the modern standards, and how we are shaping a more transparent future for clinical trials. And after Dr. Loke's presentation, Dr. Barbara Hendrickson will be presenting a pragmatic example of evaluating how well the public sources like clinicaltrials.gov, the agency summaries, journal publications, actually help us contextualize the adverse event data coming from clinical trials. Dr. Hendrickson is a seasoned PV clinician. She was the vice president of pharmacovigilance and patient safety at AbbVie, a pediatric infectious disease specialist with continued work in the University of Chicago. As Susan mentioned, she is the co-lead for the ISE workstream within the ASA, in addition to an active participant in FUSE and PSI work groups. So with that, I pass it on to Dr. Loke.
Thank you. Thank you. Very, very kind introduction. I'll just now put up my slides. Okay. Um, so my topic, which I'll spend the next 20 minutes on, is regarding guidelines or reporting or adverse effects. Why we need to do it, what do we do, and how do we do it? And we're principally talking about trial publications in this 20 minutes because we probably would have to spend many more hours if we're talking about all the other sources which all report in different ways. And that's the reason why I put up this slide, because understanding what we commonly call a safety profile is in some ways we're looking at a big beast. It's got numerous facets, and there are a lot of us working on it, and we report different things when we're looking at it, because the number of outcomes is so huge that we can't really get an easy grasp of it. And that effectively stems from the problems in clinical trials where the trial is designed to evaluate a well-defined primary outcome. It's very well specified. There's a power calculation. There's clear methods of defining and ascertaining this primary outcome, which is usually an efficacy outcome, although there are a few trials which measure harms outcomes. The, the it's quite different when you look at the huge number of adverse effects that are collected in any trial. And this creates problem for journal publications who have limited space. Each journal has varying requirements, different formats, how they want it. So the large amount of adverse effects data with diverse characteristics creates the following problems. We start with a situation where it's not easy to define or pre-specify all the adverse effects beforehand, because we know that there are unexpected ones, things that come out of the blue, things that can't be predicted. Although the there are ones that we can predict based on the known mechanism of the drug, and these can be classified usually as uh, a, serious adverse events of known interest and things like that. But there are numerous ones which come out of the blue. Because we don't know exactly what we need to monitor for, so the methods are quite variable. Obviously, you have the standard spontaneous reports thing, um, which also takes place in trials, which we would say passive surveillance. The patient can notify the investigator at any time or their clinician. Or you can have something in the protocol that says we will measure your liver function at months 1, 3, and 6. So that's active regular monitoring. And these would yield quite different answers depending which one you use.
Oh, yeah.
And we have analytic challenges, which do we lump or split? What happens when we have hundreds of adverse events and we're doing multiple testing on them? So that creates problems. And for a journal publication, the worst bit is so in that limited space, which ones do you report? What format do you put them in? Do you pull things that are in the same the same grouping? So it's quite difficult to standardize that. The bit of history behind this that is in the past, let's say we're going back 60, 70 years or whatever, we were not so concerned about this benefit harm balance because if you didn't have penicillin for pneumonia, then your outcome is obviously very poor. So we're not so worried about the adverse effects because you have a life-threatening illness that needs treatment. And certainly before we had treatment for hypertension, then people were dying of the complications with strokes and heart failure and so on. Um, but I would understand that journal articles in those days would be focusing on the benefit of this major life-saving new drugs, and harm doesn't have to be a major focus. But certainly in the past 2 or 3 decades, we have explosion of new compounds entering the market, and they're all competing in different areas, different niche. So we have primary prevention in healthy people where the adverse effects could happen tomorrow when you start the drug, but the benefit might be 10 years away. So that is a different benefit harm balance. We have drugs for less severe conditions, for example, hair loss. And we have new therapies that are approved or licensed because of perceived safety advantages or easier use, easier administration, uh, rather than being conclusively more efficacious. And this is evidenced by the number of non-inferiority trials that are coming out to show that the treatment is equally or similarly efficacious, but may have an advantage in some other way. One of the early examples is the cyclooxygenase 2 inhibitors that were aimed at replacing the traditional non-steroidal drugs. And we know how some of them, rofecoxib fell by the wayside because the safety advantage didn't materialize. And so, with the crop of compounds that we're having nowadays, the benefit harm balance considerations are completely altered from the life-saving nature of the past. And this is from 24 years ago, where a patient wrote to one of the English newspapers saying that there's a list of 83 possible side effects on that patient information leaflet, and it's utterly bewildering to the patient which ones are important. So, ba- uh, based on the example, the patient was started on a statin, and we know that a substantial number of patients don't want to take a statin because of the fear of muscle weakness, muscle pain. So we need to talk to clinicians and patients so that they understand what the exact risk is, and it allows comparisons to be made within the trial or and between trials as well, uh, with the high quality data to inform treatment decisions. Hopefully, we have a list of major and minor adverse effects that guides the use of the drugs. So, my history of getting involved in this was that um, I s- I started learning the methods of systematic reviews, and the aim was to reduce biases by using a complete data set. And we use multiple sources, corroborative evidence, and we built that in with critical appraisal of the quality so that we can identify biases. And following that, we could statistically combine the data in a meta-analysis um, to achieve a pooled estimate. So why did I enter the arena to look at systematic reviews of adverse effects and harms data? Well, I found that single trials are generally powered on an efficacy outcome. When you look at serious adverse events, they tend to, well, usually tend to have a much lower incidence rate, and therefore the the authors of the journal articles often wrote that adverse effects were not significant, whatever that means. So I decided to explore the methods of meta-analysis, that if I could pull the data from several sources, I could hopefully detect small but important outcome differences with sufficient statistical power and come up more precise estimates of the adverse effects rates. So the when I started doing this, I ran into trouble because it was hard to identify the published articles with the relevant harms data. Um, and even where the data was available, I struggled to get the relevant data out because there was a lack of detail, reporting inconsistent, and it bothered me all the time because the method section seldom described um, whether the trialists looked for specific adverse effects, which ones they looked for, and how hard did they look. And so in my reviews, I ran into these problems with data quality, which we well know if you do a review and you put rubbish in, you will get rubbish out. And it stemmed from the main problem that harms were not clearly defined, and I wasn't sure whether they were carefully monitored using sensitive and specific methods. And by this to a sensitive that it should be good enough to pick up the ones that are happening, and the specific method means that the ones that are picked up are genuinely true adverse effects rather than misclassification and so on. So, because I ran into trouble in doing the systematic reviews, I decided to look into trial publications to see whether transparent reporting was a universal feature or was it a rare occurrence. And my logic was that if I'm going to be successful in completing a systematic review and a meta-analysis, I need to be able to compare and pull the data across trials. And um, the I would say essential part of a meta-analysis is that we need standardized definitions and terms before we could even think of pulling the data. And I became aware that there are wildly varying estimates which stem from differences in monitoring. And there was one study back, I think this is from the 80s, where they said if you rely on spontaneous reports or passive surveillance, you might only have 16% of people reporting anything to the trial investigator or the clinician. Whereas if you had a questionnaire that you made them fill in, then they may report symptoms 62% of the time. So um, that created problems for me in trying to understand what the baseline rates are. And I found there's problems that there isn't a consistent numerical data, which I would like to see, which is clear numerators, clear denominators. What is the exposure periods? Um, and so when we looked at that, so this is straightforward survey. I just well, I would like to say I downloaded 185 randomized controlled trials, but back in the 90s, you couldn't download 185 RCT, so you had to get the paper versions in seven major general medical journals. And we looked at how the journal articles reported on adverse effects or adverse drug reactions. So there were 25 or 185 trial articles. We did not mention anything about adverse events, completely just absolutely nothing about it. There were many that failed to give the rates in each group um, or did not specify how they monitored for it in the methods section. Now, all of us are interested in whether they are serious or whether they're mild or moderate or severe. And unfortunately, the terminology was so heterogeneous that we couldn't really understand it because some trial articles use minor or major, some use mild, moderate, severe, some reported serious adverse events as we commonly do now. And there's the issue of space in a print journal back in 1997 that only 5.8% of the article um, section for the results and discussion was related to the harms of the intervention. So most trial reports didn't feel it worthy to spend too much space on that. So was this a one-off? And actually, it's not because a similar article in JAMA 2001 said that severity for the adverse events were defined in only 39% of trial articles. Laboratory toxicity definitions of toxicity were found in only 29%. So this creates problems because it has a negative impact on your understanding of the full safety profile. You cannot compare rates between trials because they reported it differently and they measured it differently. We couldn't judge how severe the events were. And sadly for me, I couldn't pull data in the meta-analysis to reach an overall conclusion. So because of uh, all of that work done. So there was a meeting in Montreal in 2003, which I went to, um, and that there was a group of academics, somebody from the FDA, Robert O'Neill, if anybody is old enough to remember back 2004, and journal editors, and um, so there was a reporting guideline that was already out called Consort for reporting of randomized trials, and this was considered an extension to cover definition of adverse events, how and when monitored, severity of grade, and the analytic approach. And I'll say upfront that this extension is not widely adopted. And how do we know this? This is because when we updated it to have Consort Harms 2022, we actually took a systematic look at what had um, water under the bridge in the last 20 years or so. And there was a paper reporting harms data and RCTs. Um, and yeah, we found that people don't necessarily use the consort harms extension. Um, and we had another paper in 2014, which looked at what harms outcomes are reported within the trials, and that there's clearly a selective reporting of which we are not quite sure why certain outcomes are chosen and certain outcomes are omitted. And that means we didn't get the full data set of information to understand the full thing. So, following the methods evaluation, we then went on to a Delphi consensus panel with two online rounds for scoring on items that we felt were important to list um, as a reporting guideline, and there was an in-person consensus meeting to finalize the items. And that led to the main focus of Consorts Harm 2022 was the uh, uh, emphasis, we elaborated on that importance of coding and definitions of adverse events. How do we lump them together? Um, oh, what was follow-up time? Was there censoring? Again, we talk about how and which adverse events were chosen for inclusion in the trial article and how we handled zero events. Um, Yeah, I've these are just some items of interest from the checklist because I won't attempt to reproduce the entire checklist for you, but it does say what are the specific objectives or hypotheses for the harms outcomes, or was there any specific hypothesis that's being tested? Uh, Definitions in item 6A, including how and when assessed, so that crops up again. Um, and we recognize that there are lots of spontaneous reports, unexpected ones that crop up, and they may not have been pre-specified. And so how were these identified, and who chose to analyze and report them? We got statements on recruitment follow-up, and which outcomes were omitted from the report, why it wasn't reported, and where it can be accessed. And this we can talk about later, because it is problematic to think where can we find the data subsequently. Um, yep. So just to round up in the next couple of slides. Similarly, there was a pharmaceutical industry and journal editor meeting um, where published in the BMJ, which actually reported fairly similar guidance, that there's a need to communicate most clinically relevant drug adverse event data, with the key characteristics of timing, frequency, duration, um, to use statistical analysis and therefore to avoid general text descriptions. Yeah, interesting. The last line of this recommendation says we need to maintain consistency of data across different public reports, and that is it is a problem when we got trial publications, we got clinicaltrials.gov, we got ePARS, we got drugs at FDA, and so on. Which I have personally had problems where each data source gives a different set of numbers. So, conclusions before I hand over to Dr. Hendrickson is that uh, I think among all of us, there's broad agreement on the need for improved transparency and completeness in reporting. A variety of guidelines, of which I've shown you three examples over the last 20 years. But I'm not clear that consistently good reporting has actually been achieved. And I'm hoping that we can find a way forward from here. So thank you. And we are looking at the beast again. So I'll stop sharing now.
Thank you very much, Dr. Loke, for that great presentation. So I'm going to be talking about a manuscript that was published earlier this year that's entitled Publicly Available Clinical Trial Safety Data, Review and Call for Standardization and Improved Reporting Practices. This manuscript was a collaboration between the Aggregate Safety Assessment Planning Task Force and Workstream 3, as Susan mentioned previously. So the objectives of the study were to assess the availability and transparency of clinical trial safety information from public sources. And we used 10 sample products at the time of their initial marketing approval. And we wanted to look at the utility of cited rates of anticipated serious or severe events. We use severe too, because a lot of times for oncology studies, this was more the focus. Events of interest to inform signal detection and IND safety reporting for studies in similar patient populations. So we selected 10 sample products that had been approved for different indications in the past 7 years, so fairly recently. And we focused on indications for which multiple products were approved over that time period. So we were trying to look at, studies that were more commonly being conducted in particular patient populations or disease populations. And we only included products that were approved for the indication in both the United States and the European Union. As you can see, for the public sources we were looking at, we needed to do that. And an attempt was made to include products from varying companies. So here's the 10 therapeutic indications for the products that we reviewed. You can see they included an infectious disease indication, diabetes, a couple of oncology indications, a couple of neurologic indications, and some immune-mediated disease indications. So for these 10 products, we reviewed 80 English language sources of public information. So 21 journal publications, 19 clinicaltrials.gov postings. We looked at the product approval data summaries for all of 10 of the products and drugs at FDA, and also in the European Public Assessment reports. And then we looked at the labeling, the SMPC and USPI for each of the 10 products. So the products were selected with no prior knowledge of the availability and quality of the parameters that were going to be evaluated. And the reviews focused on the pivotal clinical studies which supported the health authority source assessment for initial product approval, as described in the drugs at FDA and the ePARS. And generally speaking, that between those two regulatory authorities, the clinical studies that were mentioned were the same. So we identified the journal publications that describe the data from those pivotal clinical studies that were highlighted in the regulatory summaries and the corresponding clinicaltrials.gov postings. And all this information was evaluated by experienced safety clinical scientists and statisticians from the working group. So the parameters assessed, including the total number of participants and patient years of exposure per treatment group, the demographics, mainly the distribution of age and gender, the geography of the enrolled participants, the disease severity of the enrolled participants, which you really need to evaluate the comparability to other study populations, and then the total serious adverse event counts and frequency and confidence intervals, and the total adverse event counts and frequency. So for each of the 10 products, we also identified 4 to 5 events of interest for a total of 44 assessed across the public data sources. And we selected events of interest which were anticipated events likely to be reported as serious or severe in a trial for that study population, regardless of product exposure, or were of customary regulatory focus, like serious drug-induced liver injury. So we didn't focus on events of special interest for a particular product. We were more interested in those events that were likely to be reported as serious or severe for that particular study population in a clinical trial. So here are some examples of the selected events of interest per indication. So for diabetes, for example, one of the adverse events of interest that we picked was major adverse cardiovascular events. For psoriasis, suicidal ideation behavior, that's been traditionally a focus, especially for FDA, for approvals of products for psoriasis. Serous infections for rheumatoid arthritis, it's well known serous infections is the most common reason for hospitalization in patients enrolled in RA trials. Pneumonitis and interstitial lung disease, which is a common complication in lung cancer, and thromboembolism, which occurs at a higher rate in patients with ulcerative colitis, especially during disease flares. So for the events of interest, we looked at the total number or percentage of participants that had the event, if that was reported, as well as the event numbers. We also looked if exposure-adjusted serious or severe event rates were provided and how the events of interest were identified. Was it a single MEDRA preferred term or preferred term grouping, or was adjudication used? And if so, was the adjudication approach described? And then if a PT grouping was used, were the included PTs stated, or was an SMQ, standardized MEDRA query, or what was previously called FDA medical queries, or now called OCMQs, were those used? So now in the next couple of slides, I'll show you the results of our study. So you can see here, this graphic shows the percentage of data sources that provided information about the demographics, key demographics, the geography of enrolled participants, disease severity of the enrolled participants, and then the total number of participants per treatment group and exposure per treatment group. And the color coding gives the different public data sources. You can see here on the right. So for demographics, that could be commonly found often in journal articles, as well as the clinicaltrials.gov and the regulatory summaries. Geography of the enrolled participants was less commonly provided, but you could find that sometimes in clinicaltrials.gov or the drugs at FDA posting in particular. So for disease severity, journal articles and the regulatory summaries were the most likely to provide that information. Most of the sources gave the total number of participants per treatment group, but less commonly, the patient years of exposure per treatment group was provided. And if it was provided, it was most often in the regulatory summaries that were posted at product approval. This graphic shows the number of participants who experienced an SAE, the total SAE counts, the percentage of participants with an SAE, and the exposure adjusted rates of SAEs, and whether confidence intervals were provided. So here you can see the number of participants with an SAE and the percentage of participants with an SAE was often provided in journal articles and the regulatory summaries and clinicaltrials.gov. But the exposure adjusted rates, on the other hand, were uncommonly provided less than 20% of cases, or even less, was that information provided in the confidence interval also not so much. This graphic shows the number of percentage of participants with a serious or severe event of interest among the ones that we looked at. So you can see, you may find this information most commonly, I think, in the regulatory summaries that are posted and also potentially in journal publications, especially in the, if you find it, it's often in the supplemental tables. You'd need to access those. Sometimes you can find it in labeling. Number of serious or severe events provided for the events of interest. Generally, you're only going to find that in clinicaltrials.gov, particularly if the event of interest is represented well by one particular preferred term, because clinicaltrials.gov only gives rates of SAEs per preferred term, not any kind of grouping of, preferred terms that might represent a similar medical concept. So, if an event is well represented by one or two preferred terms, you might be able to ascertain the number of a serious or severe events from clinicaltrials.gov. But the exposure adjusted rates for these events is not commonly found. So, you can see, if you're going to find it, you might find it in journal publications or the regulatory summaries, sometimes in labeling, but it's not something that's generally found readily. So here we also show whether you could ascertain the search criteria for the events of interest, how those rates were calculated and presented. So again, the regulatory summaries are probably the best possibility of finding that information. It does show here that you might find it over 50% of the time, but I have to say, assessing whether the search criteria was provided is somewhat a subjective exercise by the experts who looked at these documents, and I think 50% is a pretty generous, I think, assessment of how often that information is provided. And I just want to show you an example from a very good journal, New England Journal of Medicine. So typically in the New England Journal of Medicine, in their adverse event tables, they label the column that gives, the medical adverse events that have been reported, they list it as event. So you don't know for sure if that's MEDRA event coding, presumably so, but it never states that at all in the paper. And then you don't know, with these adverse events, if they represent a grouping of preferred terms. They may have, as you can see here, footnotes occasionally, and I give the footnote where you can get some sense of that sometimes. But, a lot of times these appear to be grouping of preferred terms that are identified after the data is unblinded, and that the clinical trial sponsor has selected, with, I guess, judgment on their part about what events might reasonably represent that medical condition from the database that they have. So this is not going to be necessarily applicable going forward to a different database that may have other preferred terms reported that represent that medical condition. So, but you can see sometimes they do have footnotes providing some of that information, but then other times, like you can see here, anemia is listed, and we all know that anemia can be represented by multiple preferred terms of MEDRA. So here you don't know for sure what preferred terms might be included in this calculation of the rate of anemia. So in conclusion, the clinical trial safety data provided in public data sources is inconsistent But overall, the clinical summaries at drugs at FDA and the EPARs had the most information about enrolled participant characteristics, percentage of patients with an SAE, and selected events of interest. But identifying exposure-adjusted rates for these events of interest is challenging, and there's a lack of transparency regarding how those adverse events are identified for inclusion in the cited AE frequencies or incident. And information about anticipated SAEs in the study population may not be provided for particular product summaries if that was not a particular event of interest for that product. So if, maybe it is an event like for ulcerative colitis, for example, thromboembolism I mentioned, is one of the more common reported SAEs, in trials, or at least is one of interest. Yet if that wasn't a particular focus for a product, you often find no information about the rate of thromboembolism reported in the public documents for a product approved for ulcerative colitis. So overall, there's a lack of sufficient detail in public sources of clinical trial safety data that hinders their value to contextualize the rates of anticipated serious or severe adverse events in novel drug development programs and to form IND regulatory reporting decisions. So this is important because the FDA finalized their guidance on sponsor responsibilities for IND safety reporting in December of 2025. And in that document, FDA cites this trigger approach option for blinded monitoring of anticipated events in ongoing trials. But this approach requires reference exposure adjusted rates for the statistical methodologies that would be used. So if you don't have those rates, you can't really use this approach. And for a lot of companies, they don't, especially small to mid-sized companies, they don't have epidemiology groups which can look at these rates potentially in real world data sources, which requires a level of expertise as well that may not be available in a smaller company. And so they're really dependent more on public sources of information for these reference rates. And, this might not only be for blinded monitoring, but also a lot of oncology trials just have one arm and they don't have a comparator. So, having some kind of reference to be looking at that data while the trial's going on would be very useful. So at this point, we recommend for public data sources that they include the treatment exposure and patient time of follow-up, standardized definitions of exposure-adjusted incident rates, plainly indicate in the safety tables or text whether the numbers shown are MEDRA coding like SOC, like system organ class, or preferred terms, some other classification, and provide clear information about how adverse events were identified for inclusion in the quoted event frequency and incidence, and state when adjudication has been employed, and if so, provide the process details. Clinicaltrials.gov, we believe, could be very useful if the patient years of exposure for treatment groups was always provided, and a list of all reported SAEs, including total event numbers at both a PT and a SOC level, because at a SOC level may not be provided often in clinicaltrials.gov if that was given. However, assessments using PT groupings represent a similar medical concept would require converting that data from clinicaltrials.gov into a searchable format so that you could assess using, your own search criteria that you prefer, like an SMQ, apply that to that data set to get some kind of reference rate for the SAE rates. And also inclusion in drugs at FDA and EPARs of broader safety event rate information would be valuable. However, I just bring to this group's attention that the FDA, as part of their new drugs regulatory modernization initiative, posted in September 2024 that they were going to be updating their process and review template for assessment and documentation of new drug product marketing applications. And they were proposing to use an integrated review document with what they said would be succinct, integrative, focused analyses of the evidence of benefit, risk and risk management, and therapeutic individualization, like special populations. And the document would highlight key review issues that the review team thought were most pertinent to their decision-making. And this integrated review document replaced the previous documentation, which often included multiple documents authored by each discipline, like statistics and the clinical review team. But I have looked at the products, some of them, that were approved in 2025, and I looked at the integrated review document that was posted at approval of those products, and this document is a much higher level summary than previously posted, where typically information was included in multiple documents that were posted. And the amount of safety data that I saw in the ones that I looked at was very limited. And so, I think going forward, FDA's drugs at FDA posting at drug approval would not be as useful as what our study showed. But you really would have to go to the EPARs and to journal publications, I think, primarily. So I want to thank you, all the co-authors of our manuscript, which I have listed here, and I give the reference for the pharmaceutical medicine publication that came out earlier this year for anybody who wants to read it. Thank you, and I'll send this back to Ranjita.
Thank you, Barbara. And Dr. Loke, I'm impressed with the synergy or the overlapping findings that both of you independently came to. Dr. Loke looking, from an academician's standpoint, and Barbara, you looking from an industry perspective as well. And it's pretty clear that there is a pretty significant gap in this publicly available data sources in A, the availability of the data, B, the detail necessary for the individuals involved in drug development to use those data, and also a lack of consistency. So I'm going to start off with a question that Simon has. This one is for Barbara. That you mentioned that on one slide, confidence intervals for the SAEs were provided. How often are the confidence intervals reported in trials and publications? How are they interpreted? Whether for hypothesis testing and comparison or for precision of estimates. And Leon, as the statistician in the group, you're welcome to chime in as well.
Yeah, no, I'm happy to have Leanne comment on this, as well as Cindy. I don't know if I think I saw that she was on because she did that particular assessment for the publication. But I think it was pretty uncommon to find any confidence intervals. I don't know if Leanne or Cindy actually wanted to comment any further about that.
Yeah, I think it's not very common unless the safety is it's very important. May overweight the benefit, or if the sponsor want to compare with the other same class drug, so they want to show they have more safe. But those confidence interval is not for the hypothesis testing. It's really for the precision of estimations. Yeah.
Thanks, Dan. And I see Brian, you have your hand up.
Yes, hello. Yes, two very interesting presentations. Just a couple of observations. First, as to why we are where we are. First of all, I was very interested in your presentation. This what's it called? The consorts. The original publication and this recent one, because I'm with the International Society of Pharmacovigilance. It's the first I've heard of it. I've also been very much involved with SEONS and the SEON 6, and this isn't widely known. I just thought you ought to know that. So I even have not heard of it discussed at DIA at all. It's just quite interesting. Secondly, this issue of the reporting by sponsors onto public databases is not covered routinely in either GCP or PV inspections. And indeed, it's an issue for the EU pass register. As far as I'm aware, that's not routinely covered in pharmacovigilance inspections. I do believe some pharma companies, their auditors do cover clinical trial reporting, but they will only cover it in the sense, oh, have we followed the process? There is no quality check by regulators of sponsors reporting onto databases. So, to be blunt, unless there's a stick, behind this, there's, things don't happen. Surprise, surprise. Thirdly, and this is still depressing because I'm a consultant working with small companies, we have a real attitude problem still. And as you pointed out, that scientifically, there's still a lot of excitement about the benefit and getting good quality evidence. For harm, it's just compliance. It's almost bizarre. It's almost schizophrenic. The difference in attitude between R&D teams around collection of benefit evidence. Harm evidence is a compliance issue. Benefit evidence, science, getting the quality, without the lack of attention, as you say, around getting good quality adverse events. And it's just, as you say, it's this concept of the background adverse events, Barbara, of course, is very important for designing your trial and getting how many, the right numbers. And finally, of course, we've now got AI. This is now a even more critical issue, and we get this sorted out because AI is going to be, is totally dependent on good quality data sets. And this is just, what you describe, I mean, this is just very depressing because AI will just accelerate problems within the system, make matters worse unless they're being addressed at source. So I just thought I'd make those comments as more of a general pharmacovigilance practitioner. And even though I really appreciate some very good statistical comments in the chat, but these are practical points that we do need to raise within the DIA meetings, and I'll be very happy to try and take this forward with the International Society of Pharmacovigilance as well, although again we have difficulty including clinical trials within our scope of training and education.
Yeah, I might just tell a quick story and then let Leanne, I know he has some comments to make as well, but when I was at at AbbVie, I got a call one day with alarmed call from marketing because a product that had been approved, a generic product, reported that the injection site reaction rate was much lower than what was quoted in the label for our product. And they wondered, why is that? And, I quickly saw that, in the label, this other company was just reporting the preferred term injection site reaction, right? And whereas AbbVie had done a pooling of event terms representing injection site reaction, like injection site erythema, injection site induration, et cetera. And so I explained to them, you can't compare between these different public sources because they're not using the same approach to, identifying what is an injection site reaction rate. And to your point, Brian, so if you just let AI loose and say, oh, pull up, injection site reaction rates for whatever, I don't think AI is smart enough yet to know that, one source is just giving them a rate for the preferred term of injection site reaction. Another source is pooling different preferred terms to represent that medical concept. So you're just going to get gibberish, really. And that's why, it takes somebody with medical knowledge and knowledge of MedDRA to be looking through these documents to really get a sense of what's going on. So, but anyways, I turn it to Leanne. I know he had some comments to make.
Yeah, so thank you, Barbara. So I think AI could be really can help from different point of view. And one thing I'm thinking about, especially when I listen to the Dr. Lucas presentation. So despite the 20 years of guidance of the consult harm guidance, so we still saw an inconsistency for general reporting of the harms. So I think we may not be able to rely solely on the journals. We also different journals they have different template or guidance, and we may rely on different other data sources such as the clinicaltrial.gov or UJCT. They have the study protocol SAP posted, so we could leverage that to check for how the data is collected and how and how the data is groupings. But that will need a lot of effort to look at the study designs, and AI could help to synchronize all the information, and that will be very helpful for the systematic review, while we cannot find the consistency across the journal articles.
We're still waiting for questions to come up. I do have one for Dr. Yoon. So the first consort guidance came back in 2010, I believe, if I remember correctly. And then the update came in 22. And Barbara's investigative work was more recent, I think probably looking at approvals within the last 5 years or so. That was well after the consort 2022 guidelines had even come out. And we are still continuing to see this gap in the way the safety data is being reported. What do you think are the major challenges for including the consort harms 2022 checklist items in the journal publications?
Yeah, I think it's a really big problem. I think, partly as Brad Edwards has said, if you do things on the basis you're just doing a box ticking exercise, people just want to do it as quickly and easily as possible, so they don't really pay much attention or time in filling up the table that Barbara showed on the New England Journal of Medicine. I presume there was a quick data dump from somewhere that got it in. So people yeah, they consider it as less important than efficacy, so they spend less time on it. So that should we use a carrot or stick approach is the other question that Brian has already mentioned. So if a journal say, "Oh, we're not going to publish your paper unless you fulfill it," then that would be a stick approach, which I think most researchers would be there will be an uproar, I think. So would most sponsors, I think most sponsors wouldn't agree that the journal would hold such a power over them. So yeah, so I guess 2 points. The authors don't really feel they want to spend time on it. The journals don't want to enforce it because it's a pretty tough call. So we're a bit stuck on this. It's not universally accepted by both authors and journal editors. So, and that's why we can't proceed to improve the reporting. Yeah, so that's my first answer. I guess you have some more follow-up to that.
Yeah, I was just going to say, I think the other issue is that the persons who review clinical trial manuscripts for, say, New England Journal of Medicine or these major journals, no doubt they're experts in the disease a lot of times, but I doubt they're often experts in safety assessment. And many of them probably don't have an intricate knowledge of MedDRA coding and how, safety data may be collected during the course of a clinical trial and potentially assessed. So I think that's an issue, that lack of knowledge. They don't really question things on the safety data presentations. like they do probably for the efficacy assessments, and how you educate those, academic scientists about that. I don't know. It's a challenge. I mean, I'm at University of Chicago. I gave some lectures recently to our group about safety assessment of clinical trials, and they basically told me, and these are people who have been, in academics for quite a while and worked on clinical trials in some cases, uh, that they'd never heard anything. They didn't know any of what I was talking about. So, I think that's an issue for sure. And I'm not sure how to improve on that, but I think it has to come from the journal editors. And the other thing is, I think, like Brian said, if our pharmaceutical companies don't even know about the Consort 2022 checklist, we can show that to a publication to our medical writers at our pharmaceutical companies and say, do you know about this? And then try to, when, if you're in safety and you have the opportunity, or in statistics, safety statistics or other statistics, to review manuscripts from your company that are going to be submitted, you can comment that, this information is not clear, this information is not provided, and you can refer to that publication or refer to our recent publication and say, we have to do better. Even small changes could make a big difference. So, I think we all on this call could take some responsibility for that.
Cool. And Greg, thanks for posting your thoughts in chat. That the ct.gov does provide more of a standardized layout of the safety data. The challenge still remains the absence of the EAERs or the EAIRs, and that was well reflected in Barbara's data slides as well. And you recommended that for the AE groupings, a pragmatic solution would be if ct.gov provided those rates for SMQs, OCMQs, CMQs, whatever the case is. I don't know, Dr. Loke, through your outreach and your working groups, or how could one sort of pipe this up to ct.gov?
Yes, we we did have well, one of my collaborators had a PhD student who was working with ct.gov to try and I think a lot of things that we discuss here as good practice, we were trying to get ct.gov to adopt these things. Yeah, and unfortunately, because of the pandemic, that project sort of got lost and was never and never followed up. Because I think so Barbara's suggestions on how, and Greg's suggestions as well. This could be usefully uh, done with ct.gov to standardize how their reporting is, because we were dealing with anything, how do you decide which ones to put in, and this issues about preferred terms and all that, which ones can be lumped, which ones should be split, and so on. So we were working with them on that basis, but that has lapsed. I think it would be really valuable to pursue that, because I feel we've got a better chance with that rather than journal publications. Because
Agree.
there are thousands of journal editors who want different things, but if we can get the ct.gov and say, look, uh, if there was a consensus meeting on what needs to be in the table, that would be a lot better. Because I think most authors, most academic scientists are good at filling in tables. If you give them a nice template table to fill in, they got a good chance of doing that. Yeah. Thank you.
Yeah, I 100% agree that clinicaltrials.gov is our best chance to get, at least for serious adverse events, to get information about that. And if we could get clinicaltrials.gov to expand a bit the information that's there, it could be very helpful. And I think that is our best chance to get background rates is from that. But, I would say the other thing, to Greg's point, I was kind of shocked, to be honest, that, going through the documents that we did, that the number of times that SMQs were used, or they previously called FMQs or OCMQs, not very often. I mean, generally speaking, it seemed to me, especially for oncology programs, what they did was after the data was already unblinded, they looked through the data, the sponsor, and then kind of did some PT grouping based on their own judgment. That seemed overwhelmingly what was happening. And that's not very helpful, if you're trying to look for a novel program, because, a preferred term that might not have been reported in this published trial might have been reported in your trial that represents the same medical concept. So, maybe if it had been reported in the other trial, that sponsor would have included it, but wasn't reported as such. So, using SMQs or OCMQs provides a level of pre-specified groupings and standardization that would be very useful, but is not always being employed at this time, I would say.
I think that kind of goes to Richard's comment as well, that these recommendations should apply to the drug labels. Because the example that you gave, Barbara, otherwise is the downstream impact of that, where not only sponsors, but even providers and patients might be making treatment decisions based on very discrepant data. The injection site reaction group term frequency is obviously going to be much higher than a single PT. So yeah, in a way, I think in internationally, this probably needs to percolate to the labeling decision across different agencies as well.
Yeah, even for this grouping that published in the labeling or clinicaltrials.gov, so they also not consistency. So during our review in the Barbara's paper, and we also found these inconsistencies even for the same CMQs. So that really make us difficult to use those public available data source. So clearly, I think we have identified a gap. So maybe to moving forward, whether we could collaborate with academia and industry-wide, put our voice for the regulator to consider, so to improve the transparency and the inconsistencies.
And Susan also follows up with a very similar sentiment that ct.gov and the agency labeling would be more open to considering if they get a similar message from consortia, from ASA safety working group, FUSE, etc. And this webinar does kick off a good sort of consensus statement and brainstorming about and identifying this critical gap. And the question that Susan poses is, how can we work together to effect these recommended changes? And I don't know if people on the call have some ideas.
I guess I'd wonder if anybody knows how to work with clinicaltrials.gov. How would we do that to try to get their attention about trying to implement some changes? And I don't know how difficult that is. I have no idea, but I don't know where to start with that. And if anybody does, that would be great.
I can find out from the previous team if they're still around and how they managed to get in on the. It's
Thanks,
not a
Dr.
problem.
Lowe. So one thing that Brian commented was really insightful, right, about the use of AI actually compounding this problem multifold. And Barbara, from your experience, I remember having worked on this article with you, it was hundreds of man hours to collate the data that you did. And I think you had attended a conference somewhere where you had asked the question if AI might have been able to do the same exercise. I wonder if you'd be willing to share your feedback.
Oh, well, I did talk. We have at the University of Chicago, we have some persons who are experts in the use of AI for research, and you can consult them here. And I did consult one of them and talk to them about our manuscript. We had already submitted, but I said, here's what we did. Could we have done this with AI and explained some of the, you know, intricacies of the situation? And he said, no. He didn't see how we could have done it with AI, no, for some of the reasons that I stated. Yeah. He said AI just wouldn't have the knowledge to to ascertain all the nuances that we were talking about, that I explained to them. He said that no, they wouldn't have that ability to, because there's so little AI does best when things are very standardized across different kinds of sources that it's pulling information from. But if the data is so non-standardized and there's, then it's very problematic. And so that's certainly the case here, for sure. Yeah.
Great. Thank you. All right. If there are no further questions, I just want to thank Michelle for putting this webinar together. Putting something like this together takes a tremendous amount of effort. And Michelle has worked tirelessly behind the scenes to get all the speakers, the coordinators, panelists, everybody together. So I sincerely appreciate all the time that you've invested, Michelle. And with that, I want to pass it back to is it Susan or Barbara for closing comments, please?
I just wanted to make sure nobody else had any further questions before we sign off here. Any other questions? Maybe not.
Barbara did Brian Edwards. I see he has his hand up.
Yeah.
Just another interesting point which was raised at the Euro DIA, and this concerns rare diseases and orphan diseases. And there was genuine concern, particularly amongst patient groups, that will be running out of patients soon for clinical trials to get adequate numbers. And therefore, this increased the urgency around quality of data. And again, this is going to be potentially aggravated by AI, which is speeding up the identification of suitable molecules, which will be queuing up, and yet there'll be no patients to test them in. So this makes it even more critical that we've got to make every case count. No more. Do you see what I'm trying to get at? It's going to, and I think there's going to be a real attitudinal change. I think I won't be at the global DIA meeting, but this is really need to raise this topic, please.
Yeah, I would thank you, Brian. And I mean, thanks again to everybody who provided comments, Ranjita for moderating this session, and the persons who asked questions or provided comments. I think that Susan, raised the key question here is we've identified the problem, and we've even got some concrete suggestions about how to improve things. But most of us on this call are not in a position to be able to implement those changes. But what we can do, I think, as people work in pharmaceutical companies, for example, is what I said is, bring to the attention of your medical writers the consort harms checklist. If you're given the opportunity to review a manuscript that your company's putting in for possible publication, try to push to clarify some points, as we've said in our pharmaceutical medicine manuscript are often unclear. So we can raise that point when we're to our company, when we're reviewing those manuscripts. And even, like I said, small changes might make a difference. And I think that, maybe we can try, Susan, to try to get in front of FDA our concerns about this new integrated document. I mean, I can understand the rationale from FDA's point of view, but I think at least a lot of the pharmaceutical companies, when we're looking for precedent about how the safety was assessed for a product that's already been approved for a similar indication, or what the rates of certain events were in those prior clinical trials in a similar study population, that information is really very valuable to us in the pharmaceutical industry. So if that information is no longer being included in the FDA summaries, that's unfortunate. And I think, if there's a way that we can raise our concerns about that, because I think a lot of us might have missed when that document was posted for comment. So, I think that's another thing. So, those are the things I guess we can do, and trying to find, the right people at clinicaltrials.gov to reach out to and maybe initiate a discussion somehow with some of the people from the consort harms group and from this ASA Biopharma Safety Working Group, have a conversation with them if they're open to it. So these are just some particular ways I think that we could try to make a difference going forward. And obviously, when we're doing planning for studies, we need to, I'll make a plug for the aggregate safety assessment plan. Although larger companies have those plans in place, a lot of companies still don't. But in that plan, we recommend that you have how you're identifying events for review and potentially the rate calculations that you're going to be presenting going forward. And also, how are you going to be collecting high quality data? To have that all mapped out in a prospective way, is really important. And, I think some companies don't really do that until they're already in phase 3 or that's when they start doing it, when they start thinking about the integrated summary of safety. So, I mean, you got to do it a lot earlier than that to make sure that you're collecting the right data and, can analyze any signals thoroughly. So I'll make a plug, for the ASAP process, in doing that as well. I think that would help ultimately in the quality of the data, as Brian is mentioning, that we ultimately collect and report. Oh, Susan asked, no, it was that document that I mentioned that in September 2024, FDA posted on their website, that they were planning to do this updated approach with an integrated summary document. So it was posted on their website at some point, but I guess if you're not continually looking at the FDA's website for new stuff all the time, you didn't see it. So I think that, I think that's, most of us don't have people available who can look at it that frequently. So I think a lot of us missed that, which was too bad, because I think our ASAP task force would have provided comments, but by the time I happened to see it, it was past the deadline for providing comments. So I don't know if any comments were received about that posting on their website, but yeah, it'd be interesting to know if they do, because sometimes FDA does post what comments they receive from people. So I don't know if I looked, I couldn't see any follow-up about that, but that would be interesting to know if they received any comments, and if so, what the nature of the comments were.
Barbara, can you say a little bit more about the implications of how this document, I guess we don't have the exact name of it here, but how did those changes affect things for pharmaceutical companies? You talked a little bit about that. Can you just say a little bit more about the implications?
Well, granted, I didn't look at a large number, but I looked at, some of the FDA's summaries that have been posted at Drugs at FDA for products approved in 2025. And now they and the ones I looked at had this integrated summary document now instead of multiple documents. Like they used to have one for like the stats reviewers, the clinical reviewers, the pharmacology reviewers, maybe the non-clinical study reviewers. It was kind of multiple documents. And I get that maybe was not optimal in many ways, but the one thing about the clinical reviewers, if they just had a dedicated document to the clinical review, although a lot of it was, maybe a lot of efficacy, but there was more safety, and they often like took tables that presumably the sponsor provided in their submission, and they pasted them in. I know from experiences taking tables that we provided at submission. So they took, some of the tables anyways that were provided, and they put them into their clinical review. So you could see sometimes, the rates of particular SAEs, and even more, some, if the sponsor had done some kind of grouping or had looked at the rates per using an SMQ, or for example, you may see tables that the sponsor provided in their submission in that clinical review document. But at least in the ones I looked at, and I looked at a limited number, so I'm not saying that they're all like this, but in the ones I looked at, those tables aren't there anymore. There is no tables. It was just like more of a narrative review from FDA about what were the key kind of issues that they were focused on during the review of the safety. So, and again, if you're only focusing on the particular concerns for that one product, it doesn't really give you information in general about the SAE rates for events for that study population, and what might be a major focus for one product in oncology, the next product may be something different, right? So, okay, well, it gave you some information about this one event that was a concern for this product, but the next time this other product, has a different concern, and you can't find any information anywhere about that, even though it's, well known to be a complication or a frequently reported SAE in that study population. So,
Yeah,
that's kind of what
helpful.
you're up against.
I can respond to that. So the process of FDA review formatting and has evolved over time. So I joined FDA in 2018. I never did one of those reviews where they were done by each individual discipline. By the time I arrived in 2018, we were doing what are called unit reviews. We still do some unit reviews, and then we also do some integrated reviews, which is yet a different format and more streamlined, and that might be what you're referring to. I think what would be very helpful if you wanted to consider providing feedback is to consider specifically what it is that you found helpful, and provide that feedback, that that's not there anymore. And perhaps, especially if as the working group sees this as an issue, we could potentially, as a working group with members from both FDA and industry, we can maybe make some progress on that. If there's things that are no longer there, that could be helpful.
Yeah, what we could give you, we can pull from the 10 products we reviewed, the FDA summaries. We can pull examples of tables that they included in the reviews that we found particularly helpful. I guess we could do that. Yeah, because there were some tables in some of the FDA reviews that were very helpful. And so, those sorts of, and ultimately, I'm sure they were from the sponsor. They were provided by the sponsor at submission, probably. And then FDA, sometimes FDA creates their own novel tables sometimes, but I think a lot of them were probably provided by the sponsor. I know from our AbbVie submissions in the past, a lot of the tables that showed up in the FDA summaries were ones we had provided. So I
Yeah,
think that
I don't think that's done very often anymore. I think now those tables get redone within the agency.
yeah.
But probably would still have the same kind of information you would be looking for.
Yeah, so we'd have to, I don't know exactly the source of the tables that might have been useful in the summaries that we looked at in the 10 products. We looked at products that, I guess it was probably up through, might have been 2023, I think, were the product approvals we looked at. Or yeah, something like that, 2023 or 2024. So it wasn't so long ago. Yeah.
Yeah, well, let's continue that discussion offline if you think that would be productive.
Yeah. All right. Well, I think
And I apologize for interrupting your summary.
No, I didn't have anything much else to say. I just want to thank Dr. Loke for participating as an ad hoc speaker for us in this session, because when we were preparing our manuscript, we came across your publication, Dr. Loke, and realized it was highly pertinent to our manuscript. And so we cited it in our manuscript, and I want to thank you for participating today. And, providing the valuable information that you did.
Thank you for inviting me. It's a great pleasure.
Thanks.
So thanking all the participants for their interest in attending and the engagement. Brian, Simon, and everyone else who kept the conversation active and interactive for us to learn from you guys as well. With that, I pass it back to Michelle. If you want to share what the upcoming plan is for the next webinars. It's usually quarterly. I'm not sure if you have a date identified yet for the next quarter.
Yeah, Michelle, feel free. I don't think we have another one scheduled yet, but thank you for queuing that up, Ranjina, and thank you so much for your moderation today.
My pleasure. Thanks, everyone.
Bye.
Bye-bye.
Bye.
Bye, cheerio.